Hi everyone, welcome back to another episode of Neurosci IQ. In this episode, we are going to discuss the impact of coffee on our brain. Of course, as you might have already known, coffee impacts almost every organ system in our bodies, but for the purposes of this video, we will just focus on the central nervous system, particularly the brain. So the main ingredient in coffee is caffeine, and it is one of the most widely consumed stimulants for the central nervous system. It is actually metabolized by the liver, and it is converted to a lot of different substances, and there are a lot of different byproducts, but the main one or the dominant one is called paraxanthine, and paraxanthine is how coffee impacts our brain. So caffeine itself is first converted to paraxanthine, and paraxanthine is actually the substance that has the impact on our bodies. And here, as you can see, is a molecule of caffeine, and that will become important later on. So here is basically a general overview of some of the impacts of coffee or caffeine on our bodies. It is, of course, a mood elevator, makes you happy and you're less depressed or less groggy in the morning, for example. It also impacts our cognitive abilities. In some studies have shown that it increases our cognitive performance, as well as increases our attention span and alertness. So we are more awake and more ready to learn. And the next point is that it actually helps us learn through a lot of different mechanisms. But the most important one that people really care about is that it has been shown to improve our memory by a significant amount. The next one is the reflexes. And some studies have suggested that ingesting caffeine leads to better or faster reaction times or reflexes. So it might be better for athletes or other high performance activities. And these are just some of the impacts of coffee. As you might have known, coffee can have many more impacts on our bodies. So now let's get into a discussion of how it actually works. So caffeine has three primary mechanisms of action. The first one is that it is an antagonist for the adenosine receptors. It also increases the concentration of cytosol calcium levels, and it also inhibits phosphodiesterases. So if you can see, for example, just as an example, here is a distribution of dopamine neurotransmitters in the brain. And the way that, for example, coffee works is that it doesn't let the neurons to be inhibited as easily. So the neurons are more alert or more active or more ready to fire an action potential. So first, as I discussed, we will discuss the mechanism for the adenosine pathway. The adenosine is a molecule, and as you can see, here is a diagram, and it's very similar in its structure to caffeine. And adenosine binds to a wide variety of receptors, but the most important one is that adenosine in the central nervous system binds to the adenosine receptor, and it basically inhibits glutamate release, and it can also hyperpolarize neurons, so they are less likely to fire action potentials. They also, adenosine upon binding to its receptor inhibits dopamine release, as well as it functions to regulate cyclic AMP levels in our brains. So as you can see, adenosine provides a wide variety of functions, and we will see why this is very important later on. And essentially what happens is, as you can see, adenosine binds to a G-coupled receptor, and that's how it starts inhibiting the neurons. But the most important thing is, as you saw, coffee or its byproducts, metabolites, can bind to the adenosine receptor and prevent the binding of the adenosine molecule. So they basically block the function of adenosine. And if you see, for example, on the left, we have the orange adenosine molecules binding to the receptor, and the G coupled receptor becomes activated, and downstream, after many different molecules become activated, it leads to a general inhibition of neurons. And of course, it has other functions that we won't discuss in this video. However, if you see, when we ingest caffeine, its metabolites are represented by the red triangle in this diagram on the right, and it prevents the orange adenosine molecules from binding to the receptors. So the receptor remains inactivated, and we cannot activate the receptor anymore as easily. So what happens is the neurons aren't inhibited, and they are more likely to function or cause action potentials to fire. So now let's talk about the calcium mobilization that we discussed earlier in the video. So actually, this is somewhat controversial. At moderate doses, calcium is released from the endoplasmic reticulum into the cytoplasm or the cytosol. But at higher doses, some studies have actually found that calcium can be prevented from being released because of some unknown mechanism. And that's why it's kind of controversial, because it only causes calcium release at a certain threshold, and if you go past that threshold, then it might have very negative consequences on the calcium levels of the cytosol, and it could actually be toxic. So the way that this actually works is that, as you will see, it binds to the ryorhodine receptors found mainly in the nervous system, and it allows for the calcium to get released. And of course, we have many different types of RYR receptors, and the main one that is found in the brain is RYR3, and that's the one that we will focus on. And calcium has many different functions. For example, as you might have known, it's very important for synaptic transmission and neurotransmitter release. And having higher calcium levels in the cytosol allows neurons to be once again less inhibited, which makes them function more and maybe release, for example, excitatory neurotransmitters such as glutamate, and makes you more happy by releasing serotonin, dopamine, and they all increase like the happy neurotransmitters. And that's why coffee makes you more alert or more happy, because your brain is less likely to be foggy or wanting to fall asleep. And as you can see, the diagram here shows that calcium plays an essential role in signal transduction in almost all cell types, but especially in neurons. So if you see the diagram on the left, we have the RYR3 receptors being inactive. And usually, for example, a lot of the times they are inactivated, but when we ingest caffeine, as you can see with coffee, the RYR3 receptors are more likely to be opened, and when they get opened by a stimulus such as caffeine and its metabolites, they allow the calcium, which was stored in the endoplasmic reticulum, to be released into the cytoplasm. And as I discussed, the calcium ions being in the cytoplasm leads to many impacts downstream, particularly neurotransmitter release and higher rates of, for example, neuronal activation. So you get more dopamine or other neurotransmitters, as I mentioned. And here, as we already discussed previously, phosphodiesterases are enzymes that cleave the covalent bonds. And one of the main functions, there is a wide variety of them, but one of their main functions is that they inactivate cyclic AMP in different cell types, and they prevent cyclic AMP to build up in cells. Cyclic AMP has a wide variety of functions. For example, if you look at the diagram on the left, when cyclic AMP is generated or activated, it activates a wide variety of molecules such as protein kinase A, and this downstream leads to the CREB gene being activated, and then it leads to, for example, NDMA receptors and other pathways that are essential for memory formation. So essentially, cyclic AMP is very important for neuronal function, specifically learning and memory formation, while it does a wide variety of functions. And if you see the diagram on the right, caffeine and its like metabolites bind to phosphodiesterases, and they prevent them from functioning. And what happens is when phosphodiesterases are prevented from functioning, they no longer regulate or cleave cyclic AMP, which means that cyclic AMP can build up in the neurons and initiate the cascade of proteins and activate many different molecules, so your neurons are more active. And that's how caffeine does its job. By first, as we discussed, it gets converted into paraxanthine, and paraxanthine, for example, could bind to phosphodiesterases, and when and it prevents cyclic AMP from being degraded by phosphodiesterases, which leads to better memory formation and more active neurons. Thank you so much for watching this episode of NeurosciIQ. Please let us know what you think in the comments, and make sure to like and subscribe if you enjoyed. Thank you.